Good afternoon. Welcome back. So we've got our viruses inside of cells, and now we're going to go through the steps to make more viruses. And today, the next 3 sessions, we're going to talk about nucleic acids, making more nucleic acids, all right, just to put it in a little forward view. So today, we're going to start with making RNA from RNA. So this is something that happens in cells infected with RNA viruses. So just to give you a little bit of history, to put this in perspective, this is not something you would need to know, but you should understand that the discovery of RNA and how it was replicated followed a certain timeline. Remember, tobacco mosaic virus was the first virus discovered at the end of the 1800s. So quite a few people worked on it for that reason. And a guy named Wendell Stanley in 1935 was able to crystallize it, make crystals of it. And he, interestingly, found that there was about 5% RNA in the crystal. He thought this was a contaminant and that the protein was the genetic material. This is 1935, 1936. He actually got a Nobel Prize for being wrong, right? And probably this was one of the worst Nobels ever given because he absolutely thought the protein was a genetic material. Of course, it took a while for that to be shown not to be correct. Anyway, 5% RNA, and that's the genetic material. It's only 5%. In 1944, remember, DNA is shown to be genetic material. Experiment done here in New York City at Rockefeller. Avery, McCarty, and McLeod showed you could transform bacteria to have a different property using DNA. People didn't believe them for a long time. And Avery, the lead scientist, he died before they could give him a Nobel Prize. Otherwise, he would have gotten one when they finally accepted it. 1952, Hershey Chase, which you know showed DNA of a bacteriophage is genetic material. The structure of DNA solved in 1953, which is the year of my birth, by the way, so that's why I'm a scientist, I guess. 1956, the Frankel-Conrad experiment showing RNA of a virus, TMV, is genetic material. And then by 1959, we have identified many viruses that have RNA as their genetic material. And beginning in the 1960s, people are very interested in understanding how this RNA is duplicated in an infected cell because we know that cells can't copy RNA. They don't have the enzymology to do that. All right, so that's the brief history of RNA viruses. So today, we're going to talk about 3 classes from the Baltimore scheme of viruses with RNA genomes. We're going to talk about class 3, the double-stranded RNAs. We're going to talk about class 5, the minus RNAs. And then we're going to talk about class 4, the plus-stranded RNAs that don't go through a DNA intermediate. They get their own little lecture. All right, so these are, you remember the Baltimore scheme, of course. We're going to go through all of these eventually in terms of how they make the nucleic acid. Now, in the 1960s, a graduate student was working here at Rockefeller on a virus related to poliovirus. He wanted to identify something in an infected cell, an activity in an infected cell that could make RNA from RNA. His name was David Baltimore. He went on to win the Nobel Prize for discovering reverse transcriptase. And full disclosure, I did my postdoc with him. So that's why I plug him a lot, because I really enjoyed working with him, and he's brilliant as far as I'm concerned. So he did this experiment, and this is really good to understand because you're going to see from this later on why he was able to identify reverse transcriptase. So he takes cells. This is not an egg. This is a cell here on the upper left. And he infected it with, let's say, poliovirus. It was a related virus. And then he broke the cells open at different times after infection. You know, here on the graph we have 0, 1, 2, 3, 4, 5 hours. And then he added the 4 triphosphates that you need to make RNA, A, U, G, and C, and one of those would be radioactively labeled. In the ancient era, we had to use radioactivity to trace things. Nowadays, we don't have to any longer, which is good because it's a pain to get rid of, and it can be dangerous. So he then would incubate these 4 triphosphates and then measure RNA synthesis. And you can basically precipitate anything longer than about 10 bases, and you can measure that. And so what he found was the graph on the right. So first of all, he did a plaque assay. So he would take a duplicate of the cell and take the supernatant and do a plaque assay. And he would measure virus production. So the virus production is here on the right-hand y-axis. So those are PFU per mil, and those are the boxes. So you can see between 2 and 3 hours, suddenly you get a burst of virus production. There's a little bit of an eclipse period, right? During which the RNA is uncoded and the proteins are made to make new virus particles. And then it peaks at about 4 hours. This is amazingly quick replication. This is really fast. All right, so then at the same time, when you look at RNA, this is RNA polymerase activity, he called it. You can see those are the circles that between, again, 2 and 3 hours, it starts to go up, it peaks, and then it goes down. So this was the first indication that there's some enzymatic activity induced in infected cells. It's not present in uninfected cells. So it's a virus-specific activity. And this turned out to be a viral RNA polymerase. And that's a plus-stranded RNA virus that I showed you that experiment with. So that polymerase is produced when the virus infects cells. A number of years later, Baltimore and Huang found a similar RNA polymerase in the particles of minus-strand viruses. So there's a RNA polymerase in the actual particles. You could purify, in this case it was vesicular stomatitis virus, the bullet-shaped virus. You could purify the virus particles and do a similar assay with triphosphates and show there's an RNA polymerase activity in the particle. So remember that when we talk about reverse transcriptase, because his work with this made him think there must be a reverse transcriptase in retroviruses. So fast forward many years later, we start to have the ability to do genome sequences of viruses. We can translate the open reading frames, and we can say, oh, this protein looks like an RNA polymerase because it has a sequence motif. In particular, GlyAsp-Asp, GDD, is a signature for RNA polymerase. We'll see why in a moment. And then you could take that gene and express it in cells and show that you get RNA polymerase activity. And finally, we do crystal structures and solve the 3-dimensional structures of these enzymes and figure out how they work. So this is how we progress with understanding RNA synthesis. So the minus-strand RNA viruses, they have a polymerase in the virus particle. And within the particle, the RNA is coated with protein. We call it a nucleocapsid. So for example, vesicular stomatitis virus, it's a single negative-strand RNA coated with protein. We call that the nucleocapsid because it's a substructure within the membrane, within the envelope. On the right is influenza virus, which is also a negative-stranded RNA virus. Each segment, this genome is in pieces, is bound by proteins, nucleocapsid protein, and those also contain RNA polymerase. The plus-strand RNA viruses, there is no RNA polymerase in the particle. You don't need it because the plus strand, as soon as it gets into cells, can be translated and you can make the polymerase. So evolutionarily, there is no requirement to have a polymerase in the particle. So there's no RNA polymerase in plus-strand RNA virus particles. There's no nucleocapsid. There's no protein bound to the RNA, with 2 exceptions: retroviruses and coronaviruses. Retroviruses have a reverse transcriptase in the particle because they make DNA from RNA, and it has to be in the particle. And coronaviruses don't have an RNA polymerase, but their RNA is coated with protein. And you may ask, why is that the exception? Because it's a plus-strand virus, so is poliovirus. Well, we think that the RNA for coronaviruses is so long it has to be stabilized by being bound by a protein. It's much longer than that of the other viruses. So here we have poliovirus, we have flaviviruses, coronaviruses, reverse transcriptase. So polio and flavi are example where you have plus-stranded RNA, no nucleocapsid, no polymerase in the particle. Coronaviruses have a nucleocapsid, but no polymerase. And retroviruses have a nucleocapsid and reverse transcriptase in the particle. And finally, the double-stranded RNA genomes, there's an RNA polymerase in the particle. And you may remember a few weeks ago, I told you that double-stranded RNA cannot be translated. Even though it has a plus strand in it, it's hybridized to a minus strand, and ribosomes can't get to it. So it can't be translated. So that's why these viruses have to have a polymerase in the particle. So the polymerase can make mRNA, single-stranded mRNA from the double-stranded RNA, which can be translated. So the double-stranded genome RNA viruses have an RNA polymerase in the particle, but the RNA is not coated with protein. It's naked RNA, so there's no nucleocapsid. We'll talk about these in some detail today. So first of all, nucleocapsids of different viruses are shown here. We've encountered these briefly before. On the upper left, we have vesicular stomatitis virus, where the RNA, the negative-strand RNA, is bound by a single protein that is repeated over and over again, the nucleocapsid protein. That protein monomer is shown at the bottom in panel B. It has two lobes, and there's a groove in the middle that binds the RNA. So here on the left in panel A, you can see many of these nucleocapsids bound together. That's the structure of it. And if you imagine the RNA is bound in that groove, you can see how the protein basically is wrapped around the RNA in this helical structure. On the right is influenza virus, where the nucleocapsid is segmented. It's in pieces, but the same principle. You have an RNA which is coated in proteins. Here in this picture, you can see the RNA polymerase is at one end of each nucleocapsid. And similarly to the VSV nucleocapsid for influenza virus, panel G, the protein has two lobes with a groove for the RNA to fit into it. So that's what nucleocapsids look like. Now, I want to just remind you that RNA is not just a line as I draw it all the time on a piece of paper. It has secondary and tertiary structures. So it has elements such as those shown here, and I'll talk about these often because they have biological significance. Stem loop structures, where the RNA base pairs with itself from downstream to form that kind of a structure. These can be quite extensive. They can have multiple stem loops in one structure. There can be bulges, interior loops, and so forth. And these have significance because they interact with proteins of various sorts. There's also a structure called a pseudoknot, which is shown in the middle here. It happens when the loop part of a stem loop base pairs with some downstream sequence. And that results in a structure shown below there, which is, when you do the structure, the actual three-dimensional structure, which is in panel C, it kind of looks like a knot, but it's not really a knot. That's why it's called the pseudoknot. And that's going to be important for some reproductions, as you will see. But even these drawings are not accurate, right? These line drawings in green are not showing you the three-dimensional structure. So on the lower right is the leader RNA of HIV. There's the AUG where protein synthesis begins. So upstream of it, there are all these stem loops. And these actually make a very different structure when you solve the three-dimensional structure of this RNA, which is shown in the bottom here. It has a lot of space, obviously, which can't be shown simply by drawing them. So RNA structure is very important. So we have two fundamental rules for RNA synthesis. And there's an RNA in a line, which just violates what I just told you, but it's easier to do that. First of all, the genome has to be copied end to end without loss of structure. So when you make more genomes, and there are two things we're going to talk about today. We're going to talk about making genomes, and we're going to talk about making messenger RNAs. They can be different depending on the virus. So if you're copying the genome, obviously you have to copy it end to end. You can't lose any sequence, because if you do, the virus will not be able to reproduce. So copy the genome end to end, and you have to make mRNAs that can be translated by the host cell. So they have to be efficiently translated. So you have to have the right structure. An mRNA in the host cell has a cap and a poly-A tail, and viral mRNAs typically do the same. Now you're going to see today that for some viruses, the genome is the same as the mRNA, but not for all of them. All right, and we're going to go through that in some detail so you understand the distinction. There are also universal rules for making RNA, in fact, for making DNA as well, as you will see next week. And there on the top is a model of a template which is being copied by an RNA polymerase. And so the first rule is RNA synthesis begins and ends at specific sites on the template. And the polymerase sometimes can initiate de novo, which means it doesn't need a primer, or it can require a primer, which is shown in this picture. There's a primer that is base pairing with the template, and the polymerase uses the primer to start adding bases. Some polymerases need a primer, others don't. Depends on the polymerase. And you often need other viral proteins, you need cellular proteins in this process, not just the RNA polymerase by itself. RNA is made by copying the template. The polymerase sees what the base is on the template and then incorporates the complementary base. So we call that template-directed stepwise incorporation. It's elongated in a 5 to 3' direction. In other words, the new strand is made in a 5 to 3' direction, but it's reading the template in the 3 to 5' direction. So there's the template there on top, 3', 5'. And 3 and 5' refer to the chemical constituents of the RNA. And the primer initiates synthesis in this case, and we're synthesizing in a 5 to 3' direction. All right, so that's very important. In some cases, as you'll see today, there's non-templated RNA synthesis. So the polymerase can make RNA without actually copying a template. And those are very special situations that we'll give you some examples of. So let's start at the beginning with initiation. There are two general ways that you can initiate RNA synthesis. Again, you can do it de novo without a primer or primer dependent. Very simple. De novo initiation, the polymerase is able to take that first base on the template, the N1 position at the 3' end, and the right triphosphate will fit into the polymerase, and it will simply start copying. No primer needed. Now, the polymerase doesn't actually select the right triphosphate. All four go in very quickly, all the time. They're all four of them going in and out of the active site of the enzyme. And when the right one is there, then catalysis occurs. It's really an interesting procedure because, how would the enzyme know what base to, hey, give me an N, give me an A, right? No, it doesn't happen that way. They're all coming in one after another very quickly, obviously. And when the right one fits in and makes a base pair, then polymerization occurs. Primer-dependent initiation is just what it sounds like. The primer can be a protein or a nucleic acid. So here we have a protein where the first base is added to it, and that's complementary to the first base on the 3' end. And there's also examples of nucleic acid primers. In this case, it's a primer that has a cap, which is that structure at the 5' end of messenger RNAs. Now, the way that the actual catalysis works is shown here, and this is the same for both RNA and DNA. Once again, and in fact, this is a figure from the DNA synthesis chapter. So all the bases are DNA, and the nucleic acid is DNA, and there's a DNA polymerase there. We didn't change it for the RNA synthesis chapter, but it's the same. There's again our template and primer and RNA polymerase. And remember, the polymerase inserts the right base that's complementary to the template. And the way that happens is shown on the right. So we have our template strand on the very right, 3' end up here, 5' end down there. And we have the newly synthesized strand on the left. you can see we've already added a C to be complementary to that G. We have added an A to be complementary to the T. Again, if this were RNA, it would be U. And then we are in the process of adding the third base here, we being the polymerase, and this is going to happen at the active site, which you're going to see in a moment. So the T is complementary to the A. So remember, all the bases are coming in. The T goes in, and that's the one that's going to be polymerized. And what happens is this A comes in as a triphosphate with 3 phosphates, 1, 2, and 3. They're called the alpha, beta, and gamma phosphates. And the alpha phosphate carries out a nucleophilic attack on this oxygen. And that attack is positioned by these 2 magnesiums here, which are in turn coordinated by 2 aspartates, which are part of the active site of the polymerase. If you remember, the motif I told you, which is key to identify a polymerase, contains 2 aspartates, and that's where they're coming from. Those are highly conserved in the active site. You'll see exactly where they are in a moment. So you have that first phosphate doing a nucleophilic attack, helped by positioning by these magnesiums. See, this magnesium is positioning the first phosphate. The second is positioning the second and the third phosphates. And these 2 aspartates are in turn coordinating the magnesiums, holding them all into place. The 2 phosphates come off as pyrophosphate, and then a phosphodiester bond is made with that oxygen. And that's how catalysis works over and over. It's called the 2-metal mechanism of polymerase catalysis because the magnesium is a metal, and there are 2 of those that are involved in this. All right, our first question is, which is a universal rule about RNA-directed RNA synthesis? A, RNA polymerase may initiate de novo or require a primer. B, RNA synthesis initiates randomly on the RNA template. C, RNA is synthesized in a 3 to 5 prime direction. And D, RNA synthesis is always template directed, which is a universal rule. Well, most of you got the right answer, which is the first. A, RNA polymerase may initiate de novo or require a primer. Those are both correct. So it doesn't initiate randomly. It initiates at a specific place, which was on one of the slides I showed you. None of you picked that. That's good. RNA is not synthesized in a 3 to 5 prime. The template is copied 3 to 5, but the synthesis is 5 to 3 prime. And RNA synthesis is always template directed is wrong because sometimes there's non-templated synthesis, as I told you. Now, when we determine the crystal structures of the 4 classes of polymerase, we saw relationships among them. There are 4 kinds of nucleic acid polymerase. There's RNA-dependent RNA polymerase, which we're talking about today. There's DNA-dependent RNA polymerase, which makes mRNAs and tRNAs and ribosomal RNAs. And then there's DNA-dependent DNA polymerase, which reproduces our DNA and that of DNA viruses. And then finally, there is reverse transcriptase, RNA-dependent DNA polymerase. And all 4 of them are shown here. There's structures at the bottom. So CLENOW is another name for DNA-dependent DNA polymerase. T7 RNA polymerase is a bacteriophage polymerase that makes RNA from DNA. Reverse transcriptase of HIV1, that's a RNA-dependent DNA polymerase. And finally, the polio polymerase and RNA-dependent RNA polymerase. They all have a similar structure where the crystallographers say they look like a right hand where the active site is in the palm. It's shown red there. And then there are fingers and thumb domains, which sometimes the fingers and thumb touch each other as they do for the polio polymerase, but they don't for all of them. You can see the fingers and thumbs are open for the other polymerases, but they all have this right hand where the active site is in the palm. Now, if you line up their amino acid sequences, that's what's shown at top here for the 4 polymerases, you can see there are these colored regions in red and green and yellow, etc., which are highly similar. And those are the catalytically important parts of the enzyme. So for example, the yellow is right in the palm of the active site. You can see yellow in all of these. And the yellow is the part that contains these motifs that are important for catalysis. So for example, gly-asp-asp is found in the polyopolymerase, and variants of the asp-asp or asp-acin. These two residues are involved in coordinating the magnesium in the active site, because the magnesium, remember, holds the phosphates in place from the triphosphate. So that's where those are coming from. And those are right in the active site. Those are those two beta strands that you can see in the active site of the polymerase. And there are other conserved areas. For example, the green alpha helix is in many of them right there. And they have very similar amino acid sequences. So these probably all evolved from a common ancestor. I don't know what that might have been. It could have been an RNA polymerase, an RNA-dependent RNA polymerase in the RNA world, right? Because we think the RNA world came first. And maybe an RNA polymerase evolved to copy those RNAs. Initially, the RNAs could self-copy, but then a protein world emerged. I'm making big leaps here. Protein world probably took 1 billion years to get a protein world. Then we have RNA-dependent RNA polymerases. And then you probably only need a few amino acid changes to change an RNA-dependent RNA polymerase into a reverse transcriptase. And then boom, you have DNA. And now you can make a DNA world. So I think that's what happened, but I don't know if we'll ever know. So let's take a close look at the polyopolymerase to illustrate some principles. Again, this one, the thumb and fingers domain, they touch each other. So the active site is encircled. The thumb and fingers are at the top there. Here's the active site where you can see the two beta strands in yellow are the core of the active site. And those two aspartates are shown with the side chains there. And there it is, a polio sequence, gly-asp-asp, GDD. Those are those three conserved amino acids. And any of these three, depends on the virus, you can use these to say, oh, this protein might be an RNA polymerase, and do some experiments to figure that out. And so how this works is shown here now in a space-filling view. The previous view was a ribbon view. So now we've got all the atoms in there, so you can see how the RNA goes in and out. So here on the left is a top view of the enzyme. And the active site, we've cut away a little bit of the front so you can see inside. The yellow are those two beta strands in there. That's where catalysis is happening. The two residues that coordinate magnesium are shown in magenta there. Those are the two aspartates. And what happens is the template, which is a single-stranded plus RNA, goes in the top of the molecule. There's a channel in the top. It threads through the active site. It's copied, and out comes a double-stranded RNA if you think about it, it goes in the top, it curves, and then comes out the front of the molecule. So here's in the middle is a front view. You can see the double-stranded product coming out. The single strand is going in the top. It bends through the active site, comes out the front. And then in the back of the molecule, which is the third panel on the right, there's another channel which lets the triphosphates in. It's for NTP entry. So the NTPs are coming in here into the active site. And again, the yellow is those two beta strands part of the active site. So the triphosphates come in and out really quickly. And when the right one base pairs, that leads to catalysis. So the base pairing is an important part of the enzymatic reaction that would link the triphosphate to the growing chain. It's not always right. As you will see later, it makes mistakes. Sometimes it puts the wrong base in. And that's called mutation. That's why RNA viruses rule, because they make a lot of mutations. Well, without mutations, life would fail. We are mutants as well. Every cell in our body has a different DNA sequence from every other. So without mutation, life would not proceed. We couldn't evolve. So really, mutations can be good. Now, I'll show you why this polymerase prefers RNA precursors. And this is the structure of UTP bound to the polyopolymerase. We're now looking in the active site very closely. And you can see the yellow down here. That's that beta strand, part of the active site with the two aspartates here in yellow. Those are the two that are going to coordinate the magnesium. But that's not shown here. What's shown here is the molecule of UTP bound in the active site. So a UTP has come in, and here is the sugar, the ribose sugar right there, the 5-carbon ribose sugar. She's making all kinds of hydrogen bond and various interactions with amino acids in the polymerase. And a key one is this interaction of the 2' hydroxyl of the ribose with this aspartate at 238 in the red strand here. And so this is RNA. And as you know, DNA does not have that hydroxyl, deoxyribo, right? So this is the RNA base. That would be U, and this would be T at the bottom. So DNA bases cannot polymerize here because they don't form that hydrogen bond. But if you do change that one amino acid, you could get an RNA polymerase to incorporate DNA. So it doesn't take very much. So you can see how easily an RNA polymerase could evolve to do other things. So backing out now to take a bigger picture, I'm going to talk about 2 kinds of plus-strand RNA viruses, just give overviews of how they copy their genome. So we're going to talk about polioviruses, or picornaviruses and flaviviruses, and alphaviruses. And let's start with the left. These viruses have a single plus-stranded RNA in their genome, which is also the messenger RNA. So this is an example of a virus where the genome is the message. And so to make more genomes and to make more message, first you make a minus strand, and then you copy it to make more plus strands. As far as we know, that's the only role for this minus strand, just to be a template to make more plus strands. Doesn't do anything else as far as we know. All right, so let's see how that works. These viruses, this is poliovirus, now bind receptors. We've talked about that last time. The RNA comes out into the cytosol, and there it's engaged by ribosomes because it's a plus-stranded RNA. It can immediately be translated. It's translated into proteins, which are processed by a protease that's encoded in the genome, and you get structural proteins made so you can build more capsids, but you also get the RNA polymerase made so you can make more RNAs. And the RNA synthesis is catalyzed by the RNA polymerase that we've seen already. It happens on membranes. These are double vesicle or double-walled membranes that are actually induced by virus infection. They're not there before the cell gets infected. And all the RNA synthesis occurs on them. It seems to be a way of concentrating all the components in one place so that they don't have to find each other in a big cell. They're all in one place. And that plus strand gets copied to a minus, actually a double-stranded, and that gets copied to form more plus strands. So let's take a look at how this works in a little bit of detail. The genome, again, is a plus-strand RNA. It's about 7,500 bases for polio, and it has one long open reading frame that goes the whole length, which means there's a start codon near the 5' end, and then there's no stop codon until close to the 3' end of the RNA. It's almost 7,500 bases of open reading frame. So when ribosomes attach and translate this RNA, they make a long protein that's then chopped up into the final proteins by viral proteases. So these are enzymes encoded by the virus that chop the protein. And there are two, there's one called 2A and one called 3C that are responsible for all these cleavages. The RNA also has a protein at the 5' end. That is going to be the primer for RNA synthesis. Remember, I said some primers can be protein linked, and this is an example of that. At the very 5' end, there is a protein, as I've just said. It's shown here in orange. It's called VPG. It's about 22 amino acids long. It's covalently linked to the very first base in the genome, which is a U, by a phosphodiester bond. And then this is the rest of the genome. In fact, the very first 100 bases or so forms this structure, which has been called a clover leaf. 1, 2, 3, 4. I guess it's a 4-leaf clover leaf, right? 1 or maybe 3, depending on how you look at it, because there are only 3 bulges. And this is going to be important for reproduction of the genome. So here's the plus strand in its closer to its actual state. It's got some structures that are important for RNA synthesis. There's a clover leaf at the 5' end. There's our VPG protein bound to the 5' end. In the middle of the genome somewhere is a a stem loop called a cis-acting RNA element. This actually is movable. You can move it around the genome as long as you maintain the reading frame, and we'll see what that is needed for in a moment. And then at the end, there's a poly A tail and a pseudoknot. And these structures are important for getting the viral RNA replicated. Cellular mRNAs are not copied by the poliovirus RNA polymerase. Something makes the copying specific to the viral RNA, and it seems to be these structures here, the cloverleaf, the Cre, and the pseudoknot. So what happens is, in an infected cell, once you have all the proteins made, this Cre element is bound by the protease and the polymerase, a combination of the two, and it serves as a place where VPG, that primer, gets uridylated. 2 U's are added to the VPG, and that is going to serve as a primer for RNA synthesis. And this stem loop has a bunch of A's at the top, and they serve as a template for putting the U's on VPG. So this is the first step in making a primer. You put 2 U's onto VPG, which is a protein, and then it's going to serve as a primer. And here's the priming step of RNA synthesis. This actually happens on membranes, as I told you, and one of the viral protein anchors the replication complex to the membrane. So here's the viral RNA with the cloverleaf, the Cre element, and the pseudoknot there at the 3' end. There are a number of protein-protein interactions that are important to get this going. First of all, there's a poly A tail at the 3' end of the RNA. There's a cell protein called poly A binding protein, which binds the poly A, and it will also bind a molecule of polymerase that's bound to the cloverleaf. So the cloverleaf anchors the polymerase, poly A binding protein, brings the end of the RNA around, and basically makes a circular molecule. And VPG, which is uridylated at the Cre element, then base pairs with the poly A sequence, begins the priming, and copies the RNA to make a minus strand down here. So the initiation starts while the RNA is on the membrane, and it's coordinated by poly A binding protein. And then to do the other strand, you would have the same sequence of events. So poly A binding protein is important to bring the ends together, and VPG serves as the primer. So our next question is, which is a part of the poliovirus replication strategy? The production of subgenomic mRNAs. So a subgenomic mRNA is something shorter than the full-length genome, say. De novo, without primer, initiation of RNA synthesis. Circularization of template for initiation of RNA synthesis, or all of the above. So which is part of the polio replication strategy? Okay, let's see what we have here. Most of you got C, circularization for initiation of RNA synthesis. That's right, but only half of you got that. Some of you thought it was everything, or A and B. So there's no production of subgenomic RNAs for polio. It copies the whole RNA, and that's the mRNA. De novo initiation without primer. The VPG, the protein, is the primer. So we'll go back and look at this, because I obviously didn't explain it very well. Let's see. So first of all, this is the RNA genome of poliovirus. It's also the message. So there's no subgenomic mRNA. The mRNA is the message, whereas for flaviviruses, there's a smaller mRNA that is clearly subgenomic. This synthesis takes place using a primer, VPG, to which 2 U's are attached. So it's primer-dependent RNA synthesis. And the synthesis occurs via circularization of the RNA. The poly A binding protein brings the ends together. One end binds poly A, the other end binds a molecule of polymerase that's bound to the cloverleaf. Circularization of the RNA, and there you can see VPG, UU is a primer. And you know the U's are complementary to the poly A at the 3' end. Yes. Good question. Yes. How does the VPG get from there to here? It's magic. I don't show it because we don't know how it happens, right? But yes, the VPG is uridylated on the cloverleaf, which is distant from the 3' end, or maybe it's all wrapped up in some kind of structure so that everything is adjacent, but we don't really know what's going on. That's a very good question, yeah. So let's look at now an example of a plus-stranded RNA virus where there is a subgenomic RNA. Here are the flaviviruses. Maybe you've heard of chikungunya virus, which is a alphavirus that mosquito-borne that if you get it, you get terrible arthritis, and you may have it for the rest of your life. It's not in the US, but it's in many other parts of the world where Aedes aegypti and Aedes albopictus are prevalent. Anyway, plus-stranded RNA virus, it makes a minus strand so that you can make more plus strands, but it also makes a subgenomic RNA, which comes from the minus strand. And the function of this, I can't tell you. It works. They're evolutionarily competitive. It's just another strategy. So here's what happens. These viruses are taken up by endocytosis. Acidification leads to fusion of the viral and cell membranes. The RNA genome is released. The very first half of the genome is translated to form polypeptides that constitute the RNA polymerase, basically. Those are these NSP 1, 2, 3, 4. And that RNA polymerase will make a negative strand copy of the genome, and it will also make messenger RNAs from that negative strand. And so the messenger RNAs actually code for the structural proteins, the membrane proteins and the capsid proteins and so forth. This subgenomic RNA allows the coding information in the 3' half of the genome to be accessed because the translation of this RNA stops about halfway down. I think that's shown here. So here's the viral genome at the top. Right, as soon as it gets in the cell, ribosomes bind to it and they translate it, and you make the RNA polymerase. That's what these proteins are up here. So you don't need to have a polymerase in the particle, right? But you need to get these structural proteins translated. There's a stop codon at the end of the open reading frame for the polymerase. So what happens is the polymerase makes a minus strand, and then it makes an mRNA from the minus strand. And from that subgenomic mRNA, you can make all the structural proteins. I don't know the function of this apparent complexity. You know, if I were designing viruses, I would make them like poliovirus, right? Where you have a single open reading frame, boom. But I didn't design these. In fact, nature designed them, and so this apparently works, that you have a subgenomic mRNA. Now let me tell you about coronaviruses, which are quite interesting. Beyond them causing a lot of human suffering, their reproduction strategy is very different from all the others we've seen. So the RNA is a very long RNA, 30,000 bases or so. It's released upon fusion at the plasma membrane in cells that have TMPRSS2. Remember that protease that catalyzes cleavage of the spike and fusion at the plasma membrane. The RNA gets in the cytosol. It's a plus RNA, so ribosomes can engage it, and you can translate about the first half of it and make the RNA polymerase, just like with the flaviviruses. And that RNA polymerase will make negative strands, and from that make plus strands, which of course go into the assembly of new virus particles. But the polymerase will also make messenger RNAs, and it does so in a very unusual way. First, it makes minus strands complementary to the message, and then it makes from those the actual messenger RNAs that will be translated to give rise to all the proteins like nucleocapsid and spike and so forth. These messenger RNAs all come from the right-hand half of the genome. So here on the right-hand side, the messenger RNAs are shown. On the left is the open reading frame that encodes for the RNA polymerase, as I said. But in order to access the coding potential in the right-hand half of the genome, you need to make messenger RNAs. And these messenger RNAs are nested, which means they overlap, right? You can see the biggest one encodes everything all the way down to the smallest one, which just encodes one protein. So the smallest mRNA encodes the nucleocapsid. The next largest mRNA encodes nucleocapsid and M protein, but only M is made. You don't make both proteins. You only make the first protein that's encoded on each mRNA. So this largest mRNA in the top here, you will only make the 2A protein and not the others. So that's why they're called nested. And the way that they're made is really important because this is why coronaviruses, we combine like crazy. And their recombination in bats gives rise to new viruses. And that's where pandemic viruses come from. So here on the top is the coronavirus genome shown in a different way. It's still plus-stranded. We have the 5' end at the left, poly A tail at the right. And then here's an RNA polymerase beginning to copy from the 3' end. So that's a viral RNA polymerase, which is copying from the 3' end to make the anti-messenger RNA, the negative strand, right? Because this strand at the top is plus-stranded. So the polymerase starts at the 3' end. It starts to make a messenger RNA. And then what it does is the 5' end of the genome wraps around and the polymerase begins to copy it. So it gets distracted. So you can see that here in the bottom slide. The polymerase has switched from copying the plus strand at the 3' end to now copying the 5' end. And the 5' end has this little gray area, which is a leader sequence that's present on all the mRNAs. So all the mRNAs have that leader. And so these blue sequences here, TRS, are signals for the polymerase to switch to the 5' leader. And so here now would be a negative strand for one mRNA, and it would then be copied to make a plus strand, and then you have the final mRNAs, which are shown in panel F, with the leader sequence and the body that encodes the protein. This happens for each of those mRNAs. So each of these TRSs gives you a different mRNA. So you can imagine this polymerase has stopped at the first blue sequence, but if it went to the next one, it would make a slightly bigger mRNA, and the next, and so forth. So this is a form of recombination because that's what happens when the polymerase switches strands, right? It can be switching RNA from two different viruses or within the same virus here to make these mRNAs. So the polymerase has a very high propensity for recombination because of this, and that's why it very readily recombines in infected cells. So this gives you this nested set of RNAs as the polymerase goes farther and farther and farther. They all have the same 5' leader, that gray sequence, because the polymerase, after copying the body of the message, switches to the 5' end of the genome to make that leader. It's very weird. It's very weird. I wouldn't have made it this way, but nobody asked me. You know, I wasn't at the meeting where they designed this. So it works, apparently. And some of the most baroque things are seen. Okay, let's switch to negative strands. And we're going to talk about two different kinds of negative strand viruses. We're going to talk about viruses with a single molecule of RNA and viruses with a segmented genome. So we have our VSV here and influenza virus. And the strategy is very similar. So the genome is a negative strand, which is shown here, minus strand genome RNA. It's got that olive color. That has to be copied to make a messenger RNA, which is the Kelly green. But for both of these viruses, the messenger RNA is subgenomic. It's not a complete copy of the genome. So these viruses have to copy their negative strands first into a full-length plus strand, which only serves as a template for making more minus strands. Just like for the plus viruses, you go through a minus strand intermediate. For these minus viruses, you go through a plus strand intermediate. And that plus strand doesn't do anything else. It's just a template for making more minus strands. So let's start with VSV. Of course, the genome is not mRNA for VSV. It's negative stranded. So this virus brings into the cell with it an RNA polymerase, a copy of the RNA polymerase protein. And that protein, when it comes in the cell with the RNA genome, will catalyze the production of messenger RNAs. And there are multiple messenger RNAs for VSV. You can see 5 of them, 1, 2, 3, 4 of them shown here. And each of them is a subgenomic mRNA. It's not even close to being a complete copy of the mRNA. Each of those encodes a protein, either more polymerase or structural proteins that are needed to make more virus particles. And there's a key concept here I want you to think about. When the genome is not mRNA, at some point, you have to switch from mRNA synthesis to genome RNA synthesis. Okay, so initially when the minus strand comes in, you make a lot of mRNA. Why? To make proteins, because that's what you need to start reproducing. But at some point now you have to think about making new particles, and for that you need genomes. So you have to switch. And I'm going to tell you for VSV and influenza virus what causes that switch to happen. Now, VSV, again, is the bullet-shaped envelope virus with a nucleocapsid. The minus strand RNA is complex with nucleocapsid protein within the envelope. Here on the bottom is the minus strand shown, and below it are each of the messenger RNAs. There are 5 in all, 1, 2, 3, 4, 5, that encode various viral proteins, including the RNA polymerase, the spike glycoprotein, the nucleocapsid, and a few others. So these are subgenomic mRNAs. And when that viral RNA comes in the cell, that's the olive one shown second from the top, it's got a polymerase attached to the 3' end. So as soon as it gets in, it starts to make these messenger RNAs. So the messenger RNAs are shown at the top. The polymerase simply goes down, makes one, another, and it just goes all the way down the RNA and makes each of these mRNAs. But they're not full-length copies of the genome, right? So you can't use those as templates to make more minus strand. You have to make a full-length plus strand, and then from that, you make a full-length minus strand. So there's a switch from making mRNA to making full-length plus strands. A full-length plus strand does not serve as a message. It's just a template. What's the switch? The nucleocapsid protein, this N protein. So early on, when the virus first goes in, it starts to make messenger RNAs, and they can then make proteins. Eventually, after some amount of time, maybe an hour or two, you start to get more and more nucleocapsid protein made, as well as the others. And the nucleocapsid acts to switch from mRNA synthesis to full-length plus RNA synthesis. As you'll see in a moment, there is a termination event after each of these mRNAs, and the nucleocapsid antagonizes that termination so that the polymerase just keeps going. So that's shown here. So the top is the polymerase bound to the negative strand RNA. It starts to make an N mRNA, the very first mRNA at the 3' end there. And then there's a sequence, IG intergenic region, where the polymerase stops. And then it reinitiates and makes the next mRNA, and it goes all the way down the genome that way. It starts and stops and starts and stops. When you make a lot of N protein, you antagonize that process. I'm not showing it, but here when N protein is rising in concentration because you've made a lot of N mRNAs, this termination no longer occurs and you make full-length plus mRNAs. This intergenic region is also where polyadenylation occurs. So here's a close-up view of the intergenic region. The polymerase has just made a message and it's stopped. There's a termination signal in this intergenic region. And then it encounters this stretch of U's, and the polymerase begins to stutter. It just keeps churning out A's. It doesn't move. It just churns out A's until maybe 200 A's are added, and then the mRNA falls off. So now you have a polyadenylated mRNA. So that's how you get the poly A sequence. It's not actually fully encoded in the minus strand by U's. It's just 7 or so U's, and that's enough to get the polymerase to slip and start adding a lot. So that's an example of a non-templated synthesis, right? Because the whole 200 poly A is not templated in the mRNA. And again, this process, this termination, is antagonized by the nuclear protein. All right, so that's VSV. Now, next is influenza virus. Again, the genome is not mRNA. It's in pieces, but they're all negative strands, and it has to be a switch from a genome to mRNA synthesis. And so let's take a look at how that happens. Here's the genome of influenza virus, 8 pieces of negative strand RNA, each of which is bound to a protein, so it's a nucleocapsid. And each of those negative strand RNAs gives rise to the synthesis of 1 or more mRNAs, most of them one mRNA that encode a protein. And you can see at the segment 7 or 8, there's some splicing that happens to give you 2 different mRNAs from the same message. But that's not important for us now. The key is that these mRNAs, although they look like they are full-length copies of the negative strand, are not. And that's shown on this slide. So here in the middle, or the second from the top, is the negative strand RNA of each segment for influenza virus. This polymerase, which is brought in with the particle, depends on a cap, a nucleic acid cap, to initiate synthesis. That cap is actually derived from host cell mRNAs. I'll tell you in a moment how it's derived, but it's a cap which is a unique chemical structure at the 5' end of the mRNA and 10 to 13 bases, all taken from a host mRNA. It acts as a primer, and the polymerase makes a messenger RNA shown at the top. So the very first bases are host in origin because they come from the primer. And this mRNA actually stops about 20 bases short of the 5' end of the negative strand. So it's not a full-length copy. It's close, but it's not a full-length copy. And why it stops short, I'm going to show you the mechanism of that in a moment. So you make these messenger RNAs, make proteins, and at some point you have to switch to making genomes. So what do? You start making full-length plus strands, and from them you can make minus strands. So again, you have those 20 bases there that are missing. And what do you think is the switch? It's the nucleoprotein. Again, just like for VSV, when levels of the nucleoprotein rise, they cause the polymerase, first of all, to be primer independent, so you don't have any host sequence on the 5' end. But they also allow the polymerase to go all the way to the 5' end of the negative strand. Now we have a full-length plus copy of the minus strand, and that can be used to make more minus strands. So again, the switch from mRNA to genome synthesis is caused by the viral nucleoprotein. So two things I want to tell you about. First of all, the primer is very cool because it's derived from host mRNAs. There's an enzyme in the influenza polymerase. It's an endonuclease that cleaves host mRNA. So this is what a cap looks like. It's a G residue attached in a 5 to 5' linkage to the rest of the RNA. The endonuclease of the virus cleaves host mRNA, so you get cap plus 13 bases. And then these are primers for the polymerase to make a plus-stranded mRNA. So there's the 3' end of the minus strand viral RNA. This cap fragment is serving as a primer, and now you have the cap fragment joined to the new mRNA. The endonuclease is an enzyme in the viral polymerase. It is the target of a brand new antiviral called baloxavir, which just came out a few years ago. So you may have gotten Tamiflu if you had flu before. There's a new one, it's called baloxavir, which targets the endonuclease that carries out this cleavage because it's essential for RNA synthesis. The other thing I want to tell you about influenza virus is why the polymerase cannot make an mRNA that goes all the way to the 3' end of the minus strand. And that's because the architecture of the way the polymerase and the templates work. So the viral polymerase is a multi-subunit complex, 3 proteins, PB1, PB2, and PA. And one of these is the endonuclease that cleaves the mRNAs to make the primers. But what we think happens here is that the minus strand is anchored to the polymerase. So it's anchored there at the 5' end. And here is the active site down there, the red and the white. And we think that the negative strand is actually pulled through the polymerase. The polymerase isn't moving down the RNA, but the RNA is pulled through it, right? So here we're pulling the minus strand through. We're making a messenger RNA. But what happens when this loop decreases in size? Because you pulled the template all the way to the end, you can't go any further, right? Because the enzyme is not moving on the template. The 5' end is anchored. There's nowhere to go. So you start to make poly A because it just turns out that there's a stretch of U's there where the minus RNA gets stuck. So you generate a poly A tail, just very much like you did for the intergenic sequence of VSV. by the polymerase stuttering. So that's why the polymerase never gets to the end, because the way the mechanism is, the 5' end of the minus strand is anchored and you can't pull it through the active site. If it weren't anchored, you would be able to pull it through, but it's not. So you're missing those 20 bases, and you need to have a way of getting over that, which is the nucleoprotein. Question 3, how are influenza virus and VSV RNA synthesis similar? The switch from mRNA to genome RNA is controlled by an RNA binding protein. Polyadenylation occurs at a short stretch of U residues. Viral mRNAs are shorter than minus genome RNA, all of the above. All of the above is correct. All of these things are right. The switch is controlled by an RNA binding protein, the nucleoprotein, that antagonizes the termination. Poly occurs at a short stretch of U residues, and the mRNAs are shorter than the genome for both viruses. So everything is correct. Let's finish with double-stranded RNA viruses. Remember, the key here, remember, even though there's a plus strand in that duplex, ribosomes can't get to it. So we need to bring a polymerase in with the particle that can make mRNA, that can be translated, and some of those mRNAs will then be replicated to make double strands. So these viruses have segmented genomes. So you have multiple segments of double-stranded RNA. The exact number varies depending on the particular reovirus. So here's what happens, and this is going to tell you all about the double capsid. The viruses have two capsids, and they're taken up by endocytosis. Now remember, this virus does not escape the endosome before it fuses with the lysosome. The lysosome, remember, has enzymes, degradative enzymes, proteases, nucleases, etc. And this virus stays in the endosome when it fuses because those enzymes strip off the outer capsid and expose hydrophobic regions that allow the capsid to penetrate out of the endosome into the cytosol. So now you have a core, which is just the inner shell of the particle, and that core has now opened up at each 5-fold axis a pore. That's where the mRNAs are going to come out. So in the complete capsid with 2 layers, these pores are covered. We call them turrets. And then the outer shell is taken off. The virus comes out of the endosome, and now those turrets are going to be where the mRNAs come out. So within this core are the double-stranded RNAs, RNA polymerase, and the polymerase in the core begins to make messenger RNAs. And you can see them coming out of the turrets there into the cytoplasm. So the RNAs actually never leave the virus particle for these viruses. They stay inside, and that's where mRNA is made. The mRNAs come out. They're translated into protein. Those proteins can go ahead and make new virus particles containing the mRNAs and a polymerase, or multiple polymerases. Those mRNAs can be made double-stranded, and finally, the final particle can be assembled by adding the outer shell. So really unique because the mRNAs are made inside the particle, the double-stranded RNAs never leave, and you make a new particle by simply assembling around an mRNA, and then the polymerase in the particle copies the mRNA to make double-stranded RNA. So that's the genome of these viruses. There is the intact virus particle with the 2 shells, and then the core is the particle where the entire outer part has been stripped off in the endosome. And you can see these turrets are normally plugged, and those plugs are removed. And so when mRNA is made, the mRNAs can come out of these turrets into the cytosol. Now, a couple of years ago, someone did an experiment to show that, in fact, the mRNAs come out of these turrets. They took purified cores of rotavirus, which is a kind of reovirus. They incubated them with triphosphate, and they saw mRNAs coming out of each 5-fold axis of symmetry at the turrets. So apparently there's each double-stranded RNA segment is attached to the RNA polymerase via the cap, and each segment is just below the five-fold axis of symmetry so that when the mRNA is made, it can extrude out of the five-fold axis, which is what you are seeing here. And interestingly, we don't know any rotavirus or reovirus with more than 12 RNA segments. And remember, there are only 12 five-fold axes of symmetry, so that may be the limiting factor. If you attach each segment to just below the five-fold axis at the turret, you can't have more than 12. The last thing I want to tell you, which is very important, we only have 5 minutes to talk about it, unfortunately, but we'll come back to it, is that RNA viruses make a lot of mutations, and that's the origin of diversity among RNA viruses. They make mistakes. The polymerases make errors when they put bases in. They're high error rates, which can be as much as one mistake per 10,000 bases polymerized. And then that can't be corrected. So in a 10 kb virus genome, a mutation frequency of 1 in 10,000 bases means you have one mutation per genome every time the genome is copied. So you can imagine that in a cell with thousands and thousands of genomes, you can have almost every base mutated. And again, there's no error correction for this. The exception are the nidovirales, which is the order that contains the coronaviruses. They have an error correction protein, an exonuclease called XON, that corrects misincorporations. And if you take out this XON gene from a coronavirus genome, you get a 15 to 20-fold increase in mutation rate. And we think this is present in the coronaviruses because they have huge genomes, right? They have the biggest RNA genomes that we know of. And to get so long, you can't make mistakes. And so you need to have this error correction. So they have about a 10-fold lower error rate than most other RNA viruses. It's not zero, it's still higher than DNA viruses, but it allows them to exist. Now, I'll illustrate fidelity of synthesis with poliovirus. The fidelity of the polymerase is determined in the active site by how the template and the primer and the NTP interact. Remember, I told you that the NTPs come in and out very quickly. All four of them go in and out of the active site very quickly. But if it's the wrong base, the ribose will not interact with these amino acids in the active site. But if the right base is present and is base paired with the template, then you have base pairing of the ribose with the polymerase, and that allows the phosphoryl transfer. So the base pairing is essential. The right base pairing can be sensed over the wrong one most of the time and lead to polymerization. But as I said, it's not 100%. You can get one error in 10,000 bases polymerized, so it's not perfect. There is a particular amino acid on the polymerase that can be changed to make it make fewer errors. This is the polyopolymerase, the active site. There is a residue 64 on the back of the polymerase. If you change it from a G to an S, the polymerase makes fewer errors. And we think this works by slowing that conformational change where the triphosphate is sensed as being right or wrong. And so this is probably conserved in a lot of polymerases. And as you'll see when we talk about vaccines, this has some practical use. There's also another residue that affects RNA recombination. We talked briefly about RNA recombination in the coronaviruses, but in most RNA viruses, it can occur when the polymerase has two templates to pick from in an infected cell. So here are two different viral genomes in a single infected cell. The polymerase is copying one. It can then switch to copying another and make basically a hybrid molecule. That's RNA recombination. And this can happen with many plus-stranded RNA viruses, and it creates new viruses as a consequence. It can be relatively high frequency, 10 to 20% of poliovirus RNAs can recombine in a single cycle. And also on the polyopolymerase, a single residue has been identified, which, if changed, can reduce the recombination frequency. And this is located in the RNA exit channel of the polymerase, and it seems to affect the rate of the replication complex movement, but doesn't affect fidelity. So we have two amino acids on the polyopolymerase, one that affects fidelity, error rate, and one that affects recombination. So you may be asking now, why should I care about any of this? Well, all this basic molecular biology, starting from how the genome is replicated through this error rate business, has been used to engineer a new poliovirus vaccine, which is being used now, and it's been given to over 100 million kids globally. And we're going to talk about that when we talk about vaccines, because the point was to try and make a vaccine with fewer side effects. And the Cree sequence, the fidelity, the error fidelity, the recombination rate, all of this was taken into account. We'll talk about that later. So next time, we're going to then focus on mRNA synthesis for DNA viruses, transcription and mRNA processing.